like to introduce Kathryn Dalton. Dr. Kathryn Dalton is an epidemiologist, an environmental microbiologist, and veterinarian who studies environmental factors that impact health through the role of the microbiome. Her research seeks to promote One Health, the shared health of humans, animals, and the environment. She's currently a postdoctoral research fellow at the National Institute of Environmental Health Sciences at NIH. And so we are just so pleased to welcome Kathryn. Kathryn?
Hello, everyone, and greetings from North Carolina. So far in this conference, we've heard some really interesting talks about the effects of pesticides on the microbiome in soil and in the environment, and how that impacts ecosystem health. But today, I'd like to shift gears a little bit and discuss more about the role of pesticides and chemicals in general on the human microbiome and how that impacts public health. Next. Before I get started, I have a standard disclaimer that views are my own, not NIH. Next. Within this talk, I'll start by reviewing key concepts of the human microbiome and its role in health, and then delve into the role that pesticides play in this relationship. And I'd like to end with discussing future directions that I believe are needed in this field to promote public health. So we've heard talks describing the microbiome already as part of this symposium, and I'm sure many of you are already somewhat familiar with the concept of the human microbiome. It's frequently featured in the media, and there are a ton of products coming out now that claim to be good for your microbiome. But what does that actually mean? What is a healthy human microbiome? Next. Well, in general, a healthy microbiome is a diverse and balanced community of microorganisms that function as a holistic system. A diverse microbiome is both rich, or having many different kinds of species, and even, or having those species be relatively equal in abundance. This is important because each microbe will have an important function that they play in the body. And so if there are disturbances to the community, you have more microbes to fill those important roles. With regards to human health, we know that our microbiome has critical roles in immune regulation, metabolism, neurological and hormonal signaling, and overall homeostasis. Next. As such, our microbiome plays a critical role in our overall health, and disturbances to the normal balance of our microbial communities can lead to various disorders. These include gastrointestinal and neurological disorders, atopic and hypersensitivity conditions, liver, renal, and skin conditions, infectious disease risk, and many, many others, beyond what is shown in this figure. There are actually very few disease conditions that in at least some way is not related to the role of the microbiome and their microbial metabolites. And we are continually learning about new associations of our microbiome to our health. Next. Now, I want to clarify that it's not just the microbes in our gut that are related to health. While our gut microbiome is obviously a very important site, and it's probably the community most extensively studied, mostly because there are more microbes colonizing the gut compared to other sites in our body. However, any site that has even a little contact with the external environment will have microorganisms present, which will have the same key functions as our gut. This includes the respiratory tract, the upper GI tract, such as the oral community, our skin, the vaginal microbiome, plus a handful of other ones. You can see in the figure on the right that each community at a different body site, shown on the bottom axis, will have a different composition and balance of microbes. Some are listed on the side axis, where a larger circle means they are more prominent in that location. Next. Now, I want to take this one step further. It's not only the microbiome in our body that is linked to human health. As we've learned in previous talks, we are surrounded by microbes in our environment. And they are not only in the natural environment, in soil and plants, but also in the built environment, such as our homes where we spend the most time. And these environmental microbiota work to influence our health through interactions and potentially changing our own microbiome, as well as directly impacting health processes, such as in the case of antigenic stimulation and infection. Next. Adding to this overall complexity, we know that there are many preceding factors and drivers of our microbial communities, both in our bodies and in our environment. These can include lifestyle factors, such as diet, medication usage, and other people we interact with, our genetics, our early life experiences, such as mode of delivery, and our physical environment, both green spaces and adverse pollutants, including chemicals and pesticides. And so this leads us to the next section of my talk, where we'll discuss the interactions between these chemical exposures and our microbiome. Next. One factor that has been less studied in reference to human health is the complex interactions between chemical exposures and our microbiome. In fact, the National Academies put forth a report back in 2018 that emphasized this field as a key future direction needed to advance population health. Next. There is a very interesting bidirectional relationship between chemicals and microbes that I will break down. First, we are exposed to chemicals which can potentially change our microbiome. We know that various chemicals have antimicrobial properties, where they can kill select microbes. Conversely, other chemicals can promote the growth of certain microorganisms. Overall, this creates an unbalanced community, or dysbiosis. As I alluded to earlier, this will likely have consequences to the important roles that our microbiome fills in health. We have seen this in both controlled lab animal models and epidemiological studies in people from the effects of BPA, phthalates, persistent organic pollutants, endocrine disrupting chemicals like triclosan, and heavy metals on the composition of the microbiome, although most focus on the gut. Next. On the other hand, when we are exposed to these chemicals, they are metabolized by our bodies, and it's typically the metabolites that cause the toxic effects. But our microbiome plays a critical role in metabolism and can alter the absorption or metabolism of these chemicals, whether it be in our gut or oral communities for ingested chemicals, or our nasal and respiratory microbiome for inhaled chemicals. And this can potentially change the toxic effects in our body, making them more or less toxic depending on our microbial composition. So overall, our microbiome can mediate the toxic effects of chemicals. There are fewer studies in this area, most are from laboratory models, which show decreased chemical metabolites and reduced bioavailability related to the presence of certain microorganisms and microbial functional byproducts. Next. Now, with specific regards to pesticides, as that's the focus of this talk, there is still a lot we don't fully understand within this model. Next. First, we know that the microbiome itself is closely tied to our health, and changes to the microbiome alters health outcomes, as we've established in the beginning of this talk. Next. Next, it's been pretty well established that pesticide exposure leads to adverse health outcomes in both agricultural workers and the general public. I could have a whole presentation on various health outcomes in people related to pesticide exposure. I'll review some highlights from a particular study called the Agricultural Health Study, which is a prospective cohort of over 50,000 licensed pesticide applicators and their spouses in North Carolina and Iowa that have been followed for over 30 years. To date, the numerous studies coming out of this cohort have demonstrated that pesticide exposures have been linked to a whole suite of adverse health outcomes, some of which are listed here. You can see there's a big range of health effects, from cardiometabolic disorders, autoimmune conditions, degenerative disorders, respiratory conditions, birth conditions, neurological and psychiatric disorders, a range of different types of cancers, and overall increased mortality. Now, that's not to say that every pesticide causes every health effect shown here, but at least one pesticide has been linked to at least one of these outcomes, and most pesticides are linked to multiple health outcomes. Next. Lastly, and what I want to focus on, is that we also know that some pesticides have been shown to alter the microbial communities in our bodies and in our surrounding environment. However, this has been less well established than the previous 2 points, and is an area of future research growth. In controlled experiments in lab animal models, researchers have found altered gut microbiota when exposed to glyphosate and chlorpyrifos. These are the 2 studies most frequently cited. However, there have been other lab-based studies showing similar trends. Next. In human epidemiological studies, a recent report this year in the Dutch Microbiome Project of Lifetime Studies showed altered gut microbiota in occupational workers exposed to insecticide and fungicides, specifically lowered microbial diversity and increases in microbes associated with disease conditions. A birth cohort out of Brazil found changes in both maternal and infant gut microbiota related to maternal exposure of DDT and pyrethroids. Another set of studies in Washington showed changes to the oral microbiome of farm workers and their children related to blood levels of organophosphate pesticides. Next. Lastly, my colleagues and I have explored the influence of pesticide exposure on the microbial communities inside workers' homes. We found that exposure to the pesticide glyphosate in the last year, as well as past exposure to banned organochlorine pesticides such as lindane, were associated with decreased microbial diversity levels in homes. And we found specific microbes that were different in their abundance by pesticide use, specifically current use of 2,4-D and atrazine, and past use of heptachlor and lindane. The decreased microbes tended to be common soil-based ones, such as Haloterigena, while the ones that were increased could possibly be pathogenic to people, such as Fusarium and Toxoplasma. Next. However, while work in this topic is still growing, there has been no research to date that has incorporated all of these aspects and the links between them to truly understand the mechanisms of how pesticide exposures cause health effects through the pathway of the microbiome. Next. And as I discussed earlier, there is a dynamic bidirectional relationship between our microbial communities and chemicals, including pesticides, that should be considered in the context of human and also ecosystem health. No study has examined if the microbiome composition alters the toxic effects we see from pesticide exposure. Next. Additionally, we know that different pesticides have different chemical properties and cause different physiological changes in our bodies. And the combination of various pesticides may have differing effects, which may result in unique impacts on the microbiome and on our health. Next. Lastly, as we've heard from speakers in our previous sessions, and will hear from following ones, that pesticides can contain other harmful chemicals such as PFAS. Plus, we are continually exposed to these harmful chemicals like PFAS in our environment. And so the mixture of multiple chemicals and pesticides together and how they act synergistically or antagonistically in our bodies, and then how this interaction relates to changes in our microbiome, is important to assess. And so I've provided the rationale and background, as well as the current knowledge gap in relation to pesticides and our microbiome. But why is this so important to understand and address these knowledge gaps? Next. Well, I'm a researcher, so I will always argue for understanding a scientific phenomena for its own sake. But in this case, there is a direct practical application to truly defining parts of this system and how they are related. Understanding the effects of pesticides on the microbiome and vice versa can provide valuable insights into the mechanisms that underlie the impact of pesticide exposure on human health. Pesticides, like other chemicals, can be ubiquitous in the environment with widespread contact, and so can be challenging to reduce their use and exposure. If we learned anything from history, it's that even if industry does come up with alternatives to adverse products, sometimes these alternatives can also have detrimental effects that we are unaware of. So what can we do to reduce the adverse health outcomes from these widespread pesticide and other chemical exposures? Next. Well, that's where I believe the future of the field of microbiome science can come into play and be a valuable asset in potentially solving this conundrum. Buffering the microbes in our bodies and in our environment, such as our homes, can be a novel mechanism to ameliorate the toxic effects from pesticides. By creating a rich, diverse microbial community within our gut, oral, nasal, skin, reproductive sites, etc., and the microbes in our environment, our microbiome will be more resilient to changes related to pesticide exposure, and we can possibly alter metabolism of these chemicals and reduce their harmful effects. Now, this is a very novel area and approach, and there really has been no studies yet to evaluate this. Next. However, we can borrow lessons learned from the area of drug trials, where there has been a little bit of research in this area. Researchers have augmented germ-free mice with a robust microbiome and found that they responded better to the anesthetic phenobarbital and were less tolerant to opioids. I believe this data hints at the possibility of the microbiome as a future intervention point to help reduce the adverse exposure to chemicals like pesticides. But I acknowledge that this is a very future goalpost, hence why I titled this slide The Moonshot. Next. So I hope that in this talk, I've convinced you to consider the role of the microbiome when assessing the human health impacts from adverse pesticide exposure. The microbial communities, both in our bodies and our surrounding environment, perform key roles in physiology and are such intricate parts of our overall health. Hence, they can serve as valuable mediators and biomarkers to assess chemical and pesticide effects. I've proposed in this talk a sort of roadmap to investigate the possibility of the microbiome as a future intervention target to advance public health as it relates to the adverse effects that we know exist from chemicals and specifically pesticides. While there is still a lot of work to be done in this area, I believe this can be a novel and exciting field in order to protect and promote public health. Next. And so with that, I would like to thank you all for your attention, and I would be happy to take questions at this time.
All right, wonderful. Thank you so much, Kathryn. You really make a strong case for the importance of a healthy human microbiome and potential interactions with pesticides. So fascinating information. We have just a couple minutes for a couple questions. So this first question for you now, we're bringing it back to the farm community, which we've been talking a lot about in this year's conference, and just wondering, what is the strength of science to show that there's a difference between the health of farm families and farm workers using standard versus some of these more organic based, soil health based practices? Are there studies that show the difference between standard and more progressive practices on the farm and farm family health?
Yeah, that's a great question, Greg. And there absolutely has been some research in terms of different health outcomes related to whether farmers use more traditional large-scale practices versus more organic practices. And specifically, some of these outcomes do relate to the microbiological outcomes, such as the prevalence of drug-resistant bacteria, not just in the farm environment, but in the workers themselves. But as far as I'm aware, there has been no studies that have explored the effect specifically on the microbiome and how different practices and husbandry management styles impact the microbiome of not only workers, but also the greater community, the workers' families as well. But I do think that could be a very interesting avenue to further explore, because knowing that these other health outcomes we are seeing changes in, I do anticipate that we'll see changes in the microbiome as well. So that's a great question.
Okay. Future directions for research there. Okay. So one more question here. If you were to give your top couple of tips and suggestions for how we can all maintain a healthy human microbiome, what are a couple of your top suggestions?
Oh, that's a tough one. I would say we know that in general, having a diverse microbiome is beneficial. And so really just getting out there and having different experiences and exposures can be beneficial. And so particularly in early life, we know that kids that are raised on farms and these more vibrant natural environments do grow to have more robust microbiomes and are, associated with decreased diseases related to the microbiome. And so that, I think, will really help individuals out in their future. I know there's a lot on probiotics, things like that. It's a very unregulated field. So I would be hesitant to, widespread recommend probiotics. However, I think some of them are actually very beneficial. And so, yeah, I definitely recommend, particularly dietary probiotics. Whether these other topical ones, I am less certain of, but absolutely, I think that could definitely help as well, too.
Okay, great. Well, that's inspirational to us, and we can all strive for homeostasis, right? That's what we're looking for in a healthy microbiome. So thanks for those tips and all that information, future direction. So glad that we were able to pull this factor in. It's really important. So thank you so very much for joining us today and for your presentation. Once again, there are some questions in chat that we're not able to get to. So if you can visit those and provide some answers, we would appreciate that.
Will do. Thanks so much, Greg, for having me here. I really appreciate it.
Yeah, thank you, Kathryn.